Welcome to Biology at Priyajit, my dear viewers. Today we explore a key topic in cell biology, that is cell cycle regulation in vertebrates. Just as the traffic signals control the smooth flow of vehicles, the cell cycle is precisely regulated by cyclins and cyclin-dependent kinases. These molecular regulators ensure that each phase of the cell cycle occurs in the correct order and at the same right time and at the exact time. So by coordinating cell growth, DNA replication and division, cyclins and CDKs maintain normal development and prevent uncontrolled cell proliferation. So let us understand this regulatory system in a simple and systematic way. Introduction. According to Rudolf Virchow in 1855, he stated that omnicellula is cellular, that means new cells arise from pre-existing cells. So the process by which a cell divides to form new cells is called the cell division, and the definite sequence of events through which a cell passes from one division to the next is called the cell cycle. So phases of the cell cycle has basically two main phases: interphase, which is the preparatory phase. It is the longest phase of the cell cycle, and the cell grows and performs normal metabolic activities during this phase, and the events of the phases of this phase was proposed by Howard and Pelk in 1953. This phase can be divided into three main stages: G1 phase or gap 1 phase, which is responsible for cellular growth, protein synthesis. Then it is followed by S phase or synthesis phase. This phase is characterized by DNA and RNA synthesis and duplication of the centrosome, which is followed by the G2 phase, which is again another phase of cellular proliferation and protein synthesis. So after the interphase, the cell enter into the M phase or mitotic phase. So it is the actual phase of cell division. So karyokinesis occurs in this phase. Karyokinesis means nuclear division. It occurs in four stages like prophase, metaphase, anaphase and telophase, and which is followed by cytokinesis or cytoplasmic division. Division of cytoplasm occurs in the cytokinesis, and two separate daughter cells are formed in this stage. Irreversibility and external control. Basically, cell cycle events occur in a strict irreversible order and happen once per cell division. Entry into the cell cycle or cycle depends on external conditions, ensuring cells divide only when resources are sufficient. So these control prevents catastrophic errors in unicellular organisms and maintains a cooperation among cells in multicellular organisms. So what happened? The processes, the events of the cell cycle is minutely controlled. So loss of control can lead to uncontrolled division as seen in cancer. So what is the cytoplasmic regulation, which was proposed by Poturao and Johnson in 1970? Experiments fusing cells at different stages by Poturao and Johnson showed that mitotic cytoplasm contains factors that can induce premature mitosis in the interphase nuclei. The image showing their experimental conclusion. So these indicated that transitions between cell cycle stages are triggered by diffusible regulatory factors. Cell cycle checkpoints, which was proposed by Hartwell and Winnert in 1988. Checkpoints are the surveillance mechanism that halt the cycle if DNA is damaged or critical events like DNA replication or chromosome alignment are incomplete. So the checkpoints always ensure each event is completed accurately and in a proper order. And cell cycle entry that depends on the cell's internal environment. So the cell cycle is tightly controlled by external signals, regulatory factors in the cytoplasm, and checkpoints, ensuring error-free and orderly progression. CDKs and cyclin, which is the central cell cycle regulators. Cyclin-dependent kinases or CDKs are protein kinase. We know that protein kinase are the enzymes which are responsible for phosphorylating the proteins, so that drive the cell cycle transitions. CDKs are active only when it is bound to a cyclin protein. So different CDK cyclin complexes are active at specific stages. G1 cyclin that drive G1 to S phase, and S phase cyclin that initiate and maintain DNA replication. M phase cyclin that triggers mitosis. Function of the CDK cyclin complexes. When the complexes become active, CDKs phosphorylate target proteins, activating some and inhibiting others, ensuring orderly cell cycle events. Example, M phase CDK phosphorylates nuclear laminin proteins, so which induces nuclear envelope breakdown, spindle assembly, and chromosome condensation. Cyclin regulation. So how the name of the protein cyclin was deduced, because this protein cyclically upregulated and downregulated. Cyclins disappear and appear in a cyclical manner, so controlling when CDKs are active. So degradation of cyclins is controlled by ubiquitination, and the cyclins are phosphorylated, recognized by the ubiquitin enzymes, and then they are degraded. Cyclins share a cyclin box or 150 amino acids long box, which can bind with the CDK. So outside this region, sequences vary and help target specific substrates. Diversity of CDKs and cyclin. Many cells have multiple cyclins and CDKs. Example, humans. CDK1 is inducing the mitosis. CDK2, 4, 6 are responsible for G1 to S transition. And multiple CDK cyclins like DEAB regulate different phases of the cell cycle. Multiple CDK cyclin combinations then allow precise spatial and temporal control of the cell cycle and specific targeting of proteins. So the CDK cyclin system acts like a highly coordinated switchboard. Cyclins turn CDKs on at the right time, and CDKs phosphorylate the targets, and cyclins are destroyed to prevent the mistakes, ensuring the cell cycle progress smoothly and accurately. Major events and regulation of the cell cycle, with that are the key events in the cell cycle. First is the DNA replication, which happens in S phase. Phosphorylation of different proteins like nuclear laminin, which are found just beneath the nuclear membrane. So it induces the nuclear envelope breakdown at the onset of prophase. Condensing activation, which helps in chromosome condensation, which occurs and followed by occurs at started at the metaphase stage. Then unlinking of sister chromatids, anaphase separation, which induces reassembly of nuclear envelope. It is the events of telophase, which occurs at the termination of mitosis, after mitosis. So G1 phase and G1S transition. Cyclin D CDK4/6 and cyclin EA CDK2 complex phosphorylate the RB protein and then activate the E2FTP dimers, transcription of genes needed for cell cycle progression. So the inhibitors of this process is RB gene product, P16 and P27 gene products, which block progression until conditions are favorable. G1 checkpoint, they detect DNA damage via sensors. It activates ATM or ATR kinases. Control protein degradation. Cyclin D, E and P27 are degraded by ubiquitin ligase or SCF at specific times to regulate progression. So CDK2 cyclin E drives the G1 S transition. S phase, chromosome and centrosome duplication occur at the S phase. So there is a intra-S checkpoint which monitors DNA replication and stops the cycle if there is DNA damage or stalled replication forks. What is G2M transition? CDK1 cyclin B or A triggers entry into mitosis, and G2 checkpoint, that is ensure DNA replication is complete before the mitosis process. Regulation of CDK cyclin complexes. CDK cyclin complexes are tightly controlled to ensure proper cell cycle progression. So they are regulated by phosphorylation, inhibitory proteins, proteolysis, and localization. Phosphorylation, activating phosphorylation on a threonine in the T loop changes CDK shape, activates it, catalyzed by CDK activating kinase or CAC, which is responsible for this kind of activation. And inhibitory phosphorylation on tyrosine or nearby threonine inhibits CDK added by V1 kinase, which is removed by CDC25 phosphatases. Overall, CDK activity depends on these phosphorylation states, which integrate signals from checkpoints and environmental conditions. Inhibitory proteins are CKI. CKI is the kinase inhibitor, cell cycle kinase inhibitors, P16 family which binds CDKs and prevents cyclin binding, CYP family, example P27 which binds CDK cyclin complexes and blocks the activity. Most CKI act in G1S phase transition, pausing the cycle until conditions are favorable. Subcellular localization. We know that CDKs and cyclins can be kept separate from their substrates or activators. So example, cyclin B1 settles between the nucleus and cytoplasm during interphase. Nuclear accumulation of cyclin B CDK1 before mitosis ensure it can phosphorylate targets for mitotic entry. What is proteolysis? That is the degradation of proteins. I know that anaphase promoting complex which degrades mitotic cyclin allow exit from mitosis. So the CDK cyclin activity is controlled like a combination lock, phosphates, inhibitors, location and time degradation all ensure that the cell cycle proceeds only when conditions are right. Cell can withdraw itself from the cell cycle. Cells do not allow divide. So depending on conditions, they can pause or permanently stop the cell cycle. So G0 phase or quiescence phase, if conditions are not favorable, cells exit the cycle temporarily. They can re-enter G1 when conditions is improvised. Senescence, permanent withdrawal from the cell cycle due to aging or differentiation. Key checkpoint or G1 phase, the point where a cell commits to divide is called a start in yeast or the restriction point in multicellular eukaryotes. So early response genes like FOS, JUN, MEEK are activated quickly by growth factors, triggering delayed early response genes like cyclin D, and cyclin D CDK46 phosphorylates the RB protein, which releases the elongation factor E2F and ultimately activates genes needed for DNA replication, including the cyclin E synthesis. CDK2 cyclin E amplifies this signal and allowing G1S transition and DNA replication to initiate. DNA replication control, pre-replication complex or pre-RC forms at DNA origins before the S phase. CDK cyclin activity then ensures proper timing. Phosphorylates CDC6, which is responsible for inactivation of the CDK cyclin complex. Then phosphorylates MCMs, which is responsible for removal from DNA. G2 phase, it prepares for mitosis, cell growth, DNA repair, chromosome condensation. CDK1 cyclin B drives the G2 phase G2M transition. So mitotic regulation, PLKs or polo-like kinases which control mitotic entry, spindle formation and chromosome segregation. Next, there is NEMA-like kinases which assist chromosome condensation and centrosome separation. Aurora kinases that regulate multiple mitotic events, separase and securin. Separase cleaves sister chromatids and securin inhibits separase until metaphase anaphase transition. G1 arrest or DNA damage response. It inhibits G1 CDK cyclin activity. So checkpoint kinases CHK1 or CHK2 phosphorylates CDC25A, keeping CDK2 cyclin E inactive. Increase CKIs and activate P53. So ATM ATR complex activate the P53, which will activate CKI expression and stopping the cycle to allow DNA repair. So the cell cycle is carefully controlled like dominoes falling in order. Checkpoints, cyclin CDKs and mitotic kinases ensure proper DNA replication, division and response to damage. So as we begin, remember that cell cycle regulation in vertebrates is a finely tuned process. Cyclins and cyclin-dependent kinases like CDKs work together like molecular switches to ensure that each phase of the cell cycle starts and stops at the proper time, maintaining orderly cell division and genomic stability. So my dear students, keep revising, stay curious and continue exploring the wonders of cell biology. Thank you.